use a simple mechanism and a facilitated diffusion. We'll talk about that next. By the way, what is the diffusion of water called? Osmosis. This is the diffusion of water. Okay, osmosis is the diffusion of water. We're thinking about water moving across membranes here. Okay, when we talk about the diffusion of water across membranes, the movement of water across membranes helps to balance out concentrations across membranes, and it's really important in living things and in cells. So we have some special terms that we use when we talk about the diffusion of water and when we talk about how water moves across membranes. We use these terms like hypotonic solution or hypertonic solution, or we might say this is an isotonic solution. What do these things mean, and what's happening in this situation? Well, here's what we need to know about these terms, and these terms are icky, but we need to think about what's happening in a cell when it's placed in different conditions. So we've got a drawing here of 3 different cells, and they're red blood cells, and they're placed into some sort of liquidy, watery condition, right? And the little dots, so we got these red blood cells, the little dots here represent particles, and we will call these particles, I think we'll call them non-penetrating solutes. Maybe they're ions or bigger molecules like sucrose or something like that. But each cell has a lot of non-penetrating solutes in it. Like I said, ions, nutrients, things like that. Now, if we place, let's just say we place a red blood cell in pure water, 100% water, we might expect there to be a higher concentration of particles in the red blood cell than outside of the cell, right? Hardly any out here in pure water, basically none. Maybe I shouldn't even have any in this thing, okay? What about the concentration of water? Where is the concentration of water highest? Well, let's see. The concentration of non-penetrating particles or solutes is highest inside the cell. That means that there's less room essentially for water than outside the cell. The concentration of water outside the cell is going to be larger. So the concentration of water outside the cell in this diagram is higher. Which way will water tend to move? Well, it's going to move in both directions, but water's going to move down its concentration gradient into the cell for the most part. So as water moves into the cell, this cell grows, it swells, it might even burst if it's an animal cell. We like to use terms to explain, like, hey, what kind of solution has this cell been placed in? And we use one of these 3 terms over here that I've listed. The terms refer to the tonicity in a solution. The tonicity is essentially the concentration of non-dissolving solutes, or non-penetrating solutes, I should say, in a solution. And what we say here is we would say, okay, water on the outside has a much lower concentration of non-penetrating solutes than does the red blood cell. Much lower. Hypo. Hypo means lower concentration of penetrating solutes. So the solution that this red blood cell is sitting in is hypotonic to the cell. So we might say the cell is sitting in a hypotonic solution, period. The solution is hypotonic relative to the cell. Now if we put the cell in, let's say, 10% sodium chloride, that's pretty salty, all right? If we put it in 10% sodium chloride, there are lots and lots of sodium and chloride ions out here in the solution, okay? relative to inside the cell, there's way more dissolved non-penetrating solutes in the solution. What that means is the water is kind of connected to those, and there's a lower concentration of water outside of the cell than inside the cell. So water is going to diffuse by osmosis across the membrane, and it's going to primarily diffuse out of the cell. As it diffuses out of the cell is going to shrivel. We call that crenate or crenation in animal cells. Plant cells will also shrivel a little bit. And in this case, we would say, the cell is sitting in a hypertonic solution. The solution is hypertonic, meaning it has a higher concentration of dissolved particles than does the cell itself. And in this kind of a solution, the cell isn't happy at all. It shrivels. It loses water. That's not good for it. And finally, we have a condition that is maybe, 0.5% sodium chloride, let's say. And here we have a pretty much equal concentration of non-penetrating particles in the cell and out of the cell, which means we have a pretty similar concentration of water in and out of the cell. So water might move into the cell by osmosis and out of the cell at about the same rate. There's no net loss or gain of water in this particular solution. And in this situation, we would say, hey, the cell is sitting in an iso, means same, tonic solution. For animal cells, this is the happiest position it can be in, right? Animal cells like to be in isotonic solutions. Plant cells actually would prefer to be in hypotonic solutions, as a plant cell has a cell wall, and as water enters the plant cell, it enters that central vacuole and fills it up. The central vacuole starts pushing against the plant cell wall, and it won't burst. It'll just help hold up, that extra water will help hold up the plant cell and keep it rigid. Okay, what's next? Question number 3. This is just kind of a fun question. What problems does a freshwater fish face with respect to water regulation? We sometimes call that osmoregulation. That is a fish in freshwater likely to gain or lose water by osmosis?